Okay, today I'm going to talk about quality-related review considerations in teriparatide injection first generic approval. So my presentation will cover quality-related recommendations and the immunogenicity risk mitigation for teriparatide injection generic applications. I will also go over some examples of common deficiencies for NA peptide products. My presentation will help you to learn quality-related recommendations for teriparatide injection generic approval, and also some common deficiencies to highlight aspects where attention should be focused to facilitate ANDA approval. Teriparatide contains 34 amino acids, is indicated for osteoporosis. The drug product is a clear solution in a multi-dose injector pen for sub-Q injection. The reference product is from Eli Lilly with recombinant API. The first approved generic ANDAs are from Teva and Apotex. Both are Q1, Q2 the same as the RLD, and both are eligible for biowaiver, and both with synthetic API. For ANDA submitted under 505J, the applicant must demonstrate that the proposed generic drug has therapeutic equivalence with the RLD. A drug that is therapeutically equivalent also needs to be pharmaceutically equivalent and bioequivalent. I want to point out the same active ingredient is required by both PE and BE. For teriparatide injection, FDA just published the product-specific guidance in this August. Besides the Q1, Q2 and the device recommendation, this PSG also direct the applicant to follow this FDA peptide guidance for the quality considerations. In my slides, I also list those peptide products that whose PSG also reference this peptide guidance. I also list those peptide products whose PSG recommend active ingredient sameness. The FDA peptide guidance describe the comparative characterization of the following attributes for the proposed drug product and RLD. Primary sequence and physical chemical properties, secondary structure, oligomer aggregation states, biological activities, and also the use of orthogonal analytical methods is recommended. These slides provide more detail of the active ingredient characterization and comparability studies. To characterize the drug substance primary structure and physical chemical properties, the firm needs to provide evidence to ensure the identity, strength, quality, and the purity of the peptide drug substance. DMF can be used for this AI characterization. For the comparability, the firm needs to provide evidence to ensure the peptide higher order structure, oligomer aggregation profile, and biological activity in the proposed finished drug product are comparable to those of the RLD. This slide provides the examples of characterization comparability studies at the drug substance level, and also some examples of the analytical methods. To characterize the primary sequence and physical chemical properties, the firm's study needs to include primary peptide structure sequence, solubility as a function of pH, high growth capacity, log P, isoelectric point, specific optical rotation. The analytical method could be IR, MS, UV, NMR, CD, AAA, L and D amino acid content, AAS, Edman degradation, and or high resolution MS. These slides provide the examples of characterization comparability studies at the drug product level and some examples of those analytical methods. For the higher order structure, the far UV CD, FTIR, Raman can be used to characterize the secondary structure. Near UV CD, intrinsic fluorescence, DSC NMR can be used to characterize the tertiary structure. For the oligomer aggregation states, the light scattering, analytical ultracentrifugation, Jiao electrophoresis, FFF, MFI can be used to characterize reversible non-covalent soluble oligomers, and also those irreversible soluble insoluble aggregates. For the biological activities, bioidentity tests per USP monograph for teriparatide injection can be used in the therapeutically relevant bioassay. Moving on to the impurity comparability studies. Per the FDA peptide guidance, the impurity should not be greater in the test product than that in the RLD. The firm needs to conduct a comparative peptide-related impurity profiling of the RLD and the proposed generic product to identify each peptide-related impurity at level greater than or equal to 0.10%, to demonstrate the no new peptide-related impurity greater than 0.5%. For impurity found in both the proposed drug product and the RLD, the firm needs to demonstrate the level in the test product is same as or lower than that of the RLD. For impurity found at level between 0.10 and 0.5%, if not present in the RLD or in the test product at higher level than that of the RLD, the firm needs to provide justification as to why such an impurity does not affect the safety, effectiveness, or potential for immunogenicity. And also the firm should consider to use the UHPLC high-resolution MS to facilitate peak identification and ensure peak purity. Regarding the immunogenicity risk mitigation, for any new or elevated level impurities found at level of between 0.10 and 0.5%, adaptive immunogenicity risks for such impurities should be evaluated individually on the purified impurity standard. Innate immune response evaluation should be conducted on the fully formulated drug product in comparison with the ROD to ensure the generic product does not contain anything such as impurities, aggregates, contaminants, or leachables that may increase immunogenicity risk. Next, I want to go over some common deficiencies for any peptide products. The first example, the firm performed an impurity profile characterization of the test product samples aged near release using the HPLC-related substance method. FDA recommend the firm to update the study using test product samples aged at the end of the proposed shelf life and perform the characterization study using orthogonal methods that use different chromatographic separation principles and also use UHPLC high-resolution MS to facilitate peak identification and matching between the ROD and the proposed the product to characterize peak purity. The next example, the firm identified some impurities in the spec table with RRTs. FDA recommend the firm to characterize the structure of those impurities identified by RRTs as per the ANDA peptide guidance and update the spec table with identified name based on the characterization. The next example, the firm proposed a limit of no more than 0.10% for any unspecified impurity. FDA recommend the firm to revise the limit from no more than 0.10% to less than 0.10% to be consistent with the ANDA peptide guidance, which states that all impurities presented at 0.10% or greater should be identified and characterized. The next example, the limit proposed by the firm for impurity 1 is more relaxed than no more than 0.5% and not supported by the comparative RD data. FDA recommend the firm to tighten the limit consistent with and supported by the observed the RD level, or tighten the limit to 0.10 to 0.5% and supported by immunogenicity study results, suggesting the impurity 1 does not add immunogenicity risk to the product at the proposed limit. The next example, the firm reported a greater level of aggregates in the test product than those found in the ROD. The FDA recommend the firm to perform an investigation to identify the root cause and improve the product aggregation profile. The aggregation profile of the test product should be comparable to the ROD. And also the firm needs to demonstrate the capability of manufacturing drug substance or drug product, exhibit batch as necessary with acceptable control of aggregates across the proposed shelf life. Now we're moving on to the challenge question. The question number 1, consistent with the ANDA peptide guidance, when conducting a comparative impurity profiling of the generic product and ROD, identify all peptide-related impurity at a level of A greater than 0.10%, B greater than 0.5%, C greater than or equal to 0.10%, D greater than or equal to 0.1%. The answer is C. Per the FDA peptide guidance, the firm needs to identify all peptide-related impurity at a level of greater than or equal to 0.10%. Challenge question number 2. Which of the following statements is not true? A, ICH Q3A and Q3B guideline is generally applicable to all drug products, including peptide drug products. B, the limit for each peptide-related impurity can be qualified by the observed RD level for the same impurity. C, a comparative characterization of the impurity profile of the generic product and RD is recommended to establish acceptance impurity limits. D, peptide-related impurity and aggregates may pose immunogenicity risks. The answer is A. Per the ICH Q3A and Q3B, the peptide drug product are not in the scope of ICH Q3A and Q3B. In summary, my presentation provided the overview of the quality-related recommendations relevant to teriparatide injection generic approval, and also provide some, we go over some examples of common deficiencies for any peptide products. Lastly, I want to thank Cameron, Ely, and Bing for their help on my presentation. That's it. And next, please welcome Dr. Flynn.